Welcome to part 6 of the nervous system. This is the 6th in our series and the last of our nervous system screencasts. And this one is going to focus on eyes and ears, how they actually work as a part of the nervous system and allow us to detect both light and sound. So that ties back to our brain. Let's think about the structures of the brain that allow us to be able to see and to hear. Which of the lobes do you think will be most closely involved with vision? I hope you're thinking the parietal lobe and the occipital lobe. The occipital lobe is where we do our vision association, combining images and objects so that we can recognize them. And then we also process the stimulus for vision in this portion of our brain. That's called the occipital lobe. In addition, the parietal lobe is going to be involved because it is the portion of the brain where we have sensory association. And that's going to be important for vision, where we have information coming in. How about hearing? What part of your brain is going to be involved with hearing? Hopefully you're picking the temporal lobe, because that's where we have our auditory cortex, and that's going to allow us to be able to process sound information. When we look at the eye, it is a two-chambered organ. It takes in light waves and translates those light wave signals into signals that your brain can understand and recognize as images. So I'm going to show you part of this video that describes just the basic structural anatomy of the eye.
Most of the eye cavity is filled with a clear, jelly-like substance known as vitreous humor. Light from an object enters the eye, passes through the cornea, aqueous humor, lens, and vitreous humor, and is focused on the rear wall of the eye, known as the retina. The retina contains thousands of light-sensitive cells called rods and cones, which capture the object's image. These cells send nerve impulses along the optic nerve to the brain. The critical, central portion of the retina is known as the macula lutea. The macula constitutes the region of maximal visual acuity, which is used for activities such as reading, driving, and sewing.
All right, so when we look at the parts of our eye, you heard the video say that the retina is where we have our rods and cones. And rods and cones are very specialized cells that are in our eye that can receive light. They act as our receptors, and then transmission of that information to our brain will occur based on those cells being stimulated. So as light comes into our eye, then the cells that are the cones and the cells that are the rods are going to generate an impulse eventually to be sent to the brain. And so we're going to look at that process in a little more detail. But as you listen to the description, listen for how the actual mechanism works. There's a rhodopsin molecule that will be involved, and inside that rhodopsin molecule, there's an important little molecule that changes from one form of its isomer to the other, and that's what really allows us to be able to see. That's pretty amazing to me, that there's a chemical, and then inside that chemical, inside that rhodopsin, there is retinol. That light causes the change of retinol from its cis form to its trans form, and that change is what our nervous system can detect as a light stimulus. So let's look at that process a little more closely. This next little video clip is going to describe photosensitivity. I want you to listen for how an isomer helps you be able to see.
Photosensitivity in animals depends on a family of pigments called rhodopsins. In the human eye, light must travel through several layers of neural cells before it can be absorbed by the rhodopsin in rods and cones. Rods and cones are also called photoreceptors. The rod cell, named for its shape, is a modified neuron that does not produce action potentials, but it does release neurotransmitter from its base, where it forms synapses with the next neurons in the visual pathway. Each rod has an outer segment packed with membranous discs. The disc membranes are stacked inside the rod cell outer membrane. In addition to rhodopsin molecules, the disc membranes carry G proteins, also called transducin, and molecules of the enzyme phosphodiesterase. The rod cell outer membrane contains a type of sodium channel that is held open by the binding of cyclic GMP molecules. The open sodium ion channels allow sodium ions to flow spontaneously from a higher sodium ion concentration outside the cell to a lower concentration inside the cell, thereby depolarizing the cell. Light sensitivity begins with the absorption of light by rhodopsin. Rhodopsin consists of a protein part called opsin and a light-absorbing part called 11-cis-retinal, which is cradled in the center of the opsin and bound covalently to it. When 11-cis-retinal absorbs a photon of light energy, the molecule rotates around an internal bond, straightening out, and becomes all trans-retinal. The shift in retinal forces a conformation change in the opsin that signals the detection of light. The photoexcited rhodopsin activates transducin by stimulating it to release GDP and pick up GTP. Activated transducin, in turn, activates phosphodiesterase. Activated phosphodiesterase then converts cyclic GMP to GMP. The reduction in cyclic GMP inside the cell allows the sodium channels to close. Pumps in the cell membrane continuously work to drive sodium ions out of the cell, so the cell can now become more negative. The membrane hyperpolarizes. Signal transduction cascades have enormous amplification abilities. In this example, each molecule of photoexcited rhodopsin can activate several hundred transducin molecules, thus activating a large number of phosphodiesterase molecules. The catalytic capacity of phosphodiesterase is great. One molecule can hydrolyze more than 4,000 molecules of cyclic GMP per second. Thus, a single photon of light can cause a huge number of sodium channels to close. To test how a rod cell responds to light, we can penetrate a single rod cell with an electrode and record its membrane potential in the dark and in the light. In the dark, the membrane potential is around minus 35 millivolts. This value is considered depolarized, that is, relatively close to zero. In the dark, when the cell is depolarized, it continually trickles out neurotransmitter from its synaptic terminals. A brief light stimulus triggers the membrane to get more negative, that is, to hyperpolarize. As the membrane hyperpolarizes, the cell releases less neurotransmitter onto the next neurons in the visual pathway. A brighter light stimulus results in an even stronger hyperpolarization and an even greater reduction in neurotransmitter release. Note that this is the opposite of what other types of sensory receptors do. They typically depolarize and release neurotransmitters in response to a stimulus. To be a system sensitive to light changes, the signal transduction cascade needs to turn off soon after the signal has stopped. With vertebrate eyes, the retinal and the opsin separate from each other soon after light absorption, causing the molecule to lose its photosensitivity. A series of enzymatic reactions is then required to return the all-trans retinal to the 11-cis isomer, which then recombines with opsin so that it once again becomes the photosensitive pigment rhodopsin. Additionally, the G protein has an automatic shut-off mechanism. After a short while, it cleaves its GTP to form GDP and thereby becomes inactive again. Without the activated G protein, the phosphodiesterase stops its activity, while another enzyme in the cell replenishes the supplies of cyclic GMP. The sodium channels open again, bringing the cell to its resting state.
So our ability to see is dependent upon a stimulus, in this case light, coming in to be received by a receptor. We have two types of receptors, rods and cones, and those receptor cells then are going to send that sensory information along a sensory neuron to the brain. The part of the brain that receives this sensory information is going to be the parietal lobe, and then we make sense out of what we're seeing with that visual association that is going to occur in the occipital lobe of our brain. And from the input that we get, this processing or integration that is occurring in the brain allows us to have vision or sight, because our brain is making sense out of the stimulus information that it receives, and that allows us to have that perception of vision. Now, you probably have heard of a blind spot in a person's eye, and that has to do with the fact that right in the back of the eye, where the conduction is going to occur, then we end up having a blind spot because the point at which that nerve leaves the back of the eye, we are then unable to detect any additional light in that particular spot, and so it shows up as a blind spot. The ability of humans to see things that are near or far has to do with the actual shape of the lens. Notice when we are looking at things that are close to us, that the ciliary muscle is contracted. It actually changes the shape of the lens. It gets more spherical in order for that light to hit the retina at the exact right spot and have the focal point actually reach the back of the eye. And then when we're seeing in distance, we're looking at something that's far away, the ciliary muscle actually relaxes, and the change in the shape of the lens makes it become a little more oval, and so we're able to process distance vision as well as light that's being reflected off things that are close to us. And so we have great variation in our capacity or ability to see things close or far away because of the shape change that occurs in the lens. Our next receptor that we want to consider today is the human ear. And the human ear is known for having three main parts, and that ear then can take sound waves and translate them into signals, and those signals then would be processed by the brain. And remember, we said that that was the temporal lobe of the brain that's actually responsible for processing input from the human ear. So when we consider the components of the human ear, there's the outer, middle, and inner ear, each of them having a slightly different role in our ability to hear. So when we are going through the process of hearing, what happens in the areas, each of them are numbered here, and so we'll trek across the ear and show how we actually can hear. At area 1, you see sound waves are coming into the ear. The ear is built, it has a specific shape that allows us to receive those sound waves. So at point number 2, you see those sound waves are sort of being funneled by this outer ear, and they're going to be moved into the ear canal. And as those sound waves move in the ear canal, it's going to bump against the tympanum or the eardrum, shown here at number 4. As that eardrum starts to vibrate, then the vibrations are going to move the tiny bones that are inside the ear, and those bones are going to carry the vibrations deeper into the inner ear and into those fluid-filled tubes called the cochlea. The fluid inside the cochlea then is going to vibrate, and a series of little tiny hairs that are inside there are going to be stimulated, and they are attached to the auditory nerve. And so that auditory nerve then is going to send the signal to the brain, and the brain processes those signals as sound. And so that's the basics of how we hear. So let's look at that process in a little more detail.
The ear is responsible for translating variations in air pressure, whether from speech, music, or other sources, into the neural activity necessary for our perception and interpretation of sound. The ear can be divided into 3 principal sections: the outer ear, the middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear, or pinna, and a canal that leads to the eardrum. The configuration of the external ear amplifies sound, particularly at the frequency ranges of 2000 to 5000 Hz. Not coincidentally, most human speech sounds are distributed in the frequency range around 3000 Hz. From the ear canal, the sound waves vibrate the eardrum, which in turn vibrates three tiny bones in the middle ear, the malleus, incus, and stapes. The stapes vibrates a small membrane at the base of the cochlea, the oval window, which transmits amplified vibrational energy to the fluids of the cochlea. The round window serves to dampen the movement of fluid within the cochlea. The complex structures of the inner ear convert sound into neural activity. In mammals, the auditory portion of the inner ear is a coiled structure called the cochlea. The region nearest the oval window membrane is the base of the spiral. The other end, or top, is referred to as the apex. Along the length of the cochlea are three parallel canals: the tympanic canal, the vestibular canal, and the middle canal. The principal elements for converting sounds into neural activity are found on the basilar membrane, a flexible structure that separates the tympanic canal from the middle canal. Let's take a closer look at the basilar membrane by unrolling the cochlea and peering inside. The basilar membrane is about 5 times wider at the apex of the cochlea than at the base, even though the cochlea itself narrows toward its apex. It vibrates in response to sound transmitted to the fluid-filled cochlea by deflections of the oval window initiated by the bone of the middle ear. High-frequency sounds displace the narrow, stiff base of the basilar membrane more than they displace the wider, more flexible apex. Mid-frequency sound maximally displace the middle of the basilar membrane. Finally, lower frequency sounds maximally displace the apex. Within the middle canal and atop the basilar membrane is the organ of Corti, the collective term for all the elements involved in the transduction of sounds. The organ of Corti includes 3 main structures: the sensory cells, or hair cells, an elaborate framework of supporting cells, and the terminations of the auditory nerve fibers. The heights of the stereocilia increase progressively across the hair cell, so the tops approximate an inclined plane. Atop the organ of Corti is the tectorial membrane. The stereocilia of the outer hair cells extend into the indentations in the bottom of the tectorial membrane. The movements of fluid in the cochlea produce vibrations of the basilar membrane. These vibrations bend the stereocilia inserted into the tectorial membrane. Depending on the direction of the bend, the hair cells will either increase or decrease the firing rate of auditory nerve fibers.
So we have looked at 2 very specific examples of sensory reception in the human body. We considered the eye and the ear. So I'd like for you to find a space on your note page to make a table that you can use to summarize your learning today. When we consider the eye, then I want you to record what the stimulus is, the sensory receptor, the sensory neurons, the integration, and then of course that response. And hopefully your summary looks like this: that the stimulus for the eye is of course light waves. It is received by the sensory receptors, in this case the rods and cones. The sensory neurons go together to form the optic nerve. Integration of this input is going to occur in the occipital and parietal lobes of the brain, and our result is going to be that we have vision. Let's do the same thing for the ear. Take a moment to fill out what you think each of these would be: the stimulus, the receptor, the neurons, the integration, and the response for the ear. And hopefully you have that the sound waves serve as stimulus for the ear, and then the basal membrane inside the cochlea is our sensory receptor for the most part. And then the sensory neuron is going to be the auditory nerve. That information is going to be taken to the temporal lobe of the brain, where it's going to be integrated or processed, and then that ends up with us being able to have hearing. So those are two distinct examples of how our nervous system can receive sensory information, then integrate and formulate a response to that. And that wraps up our exploration of the nervous system.